After the synthesis of the DNA, that is the process of replication, the synthesis of RNA molecule from the DNA is a complex process involving one of the group of RNA polymerase enzymes and number of associated proteins. General steps are the same as required to synthesize the primary transcripts, which are initiation, elongation, and termination. So, a number of DNA regions and protein factors that bind to these sequences along to regulate the initiation of transcription factors have to be studied first. So, before going into the details, all eukaryotic cells have four major classes of RNAs: ribosomal RNA, messenger RNA, and transfer RNA, and some are small nuclear RNAs as well. So, RNA is synthesized from DNA template by an RNA polymerase. The process of DNA and RNA synthesis are similar, and they involve the general steps of initiation, elongation, and termination. But these processes differ in several important ways, including: first of all, the ribonucleotides are used in RNA synthesis rather than deoxyribonucleotides. Secondly, uracil replaces with the thymine as the complementary base pair of adenine in RNA. And one of the most important difference between DNA and RNA synthesis is that the primer is not involved in the synthesis of RNA. Only a very small portion of genome is transcribed or copied into RNA, where the entire genome must be copied during DNA replication. And last but not the least, there is no proofreading function during the RNA synthesis. The process of formation of transfer RNA, ribosomal RNA, and messenger RNA is called as transcription. So, DNA is responsible for the synthesis of messenger RNA, transfer RNA, ribosomal RNA, and small nuclear RNAs. We will discuss this transcription process both in eukaryotes as well as in prokaryotes. So, we're gonna start the transcription process in prokaryotes, which includes first of all the initiation, which is done with the help of RNA polymerase. The DNA dependent RNA polymerase, which is known as RNAP, from the bacterium E. coli, is a very complex structure, just like the diagram which is from your Lippincott 30.6 shows the structure of RNA polymerase. It has a complex consisting of two identical alpha subunits, similar but not identical beta and beta dash subunits, and omega subunit. Beta is thought to be a catalytic subunit. So overall it is called as a metalloenzyme. And the sigma part of this RNA polymerase is identical unit, which when in combination with the other parts of the RNA polymerase is called as a holoenzyme. So we can say that holoenzyme is equal to sigma factor, which is the upper mouth of the RNA polymerase, plus all the other 5 subunits. Secondly, what this RNA polymerase is going to do, it recognizes the promoter regions. So this is another important term which you're going to learn in this lecture, is the promoter. What basically the promoter is, this is the conserved sequences which are present in all the species and responsible for the recognition of initiation machinery. So the initiation machinery in the RNA synthesis is RNA polymerase. So this RNA polymerase goes and recognizes promoter recognition sites. So this RNA is, you can say that this RNA is specific for promoter recognition specificity. And then there must be, after the recognition, we will go towards we will move towards the second step, which will be the elongation. During elongation, a single stranded RNA will be protruding out from the double stranded DNA. So in the overall process, one of the strand of the DNA could be called as a coding strand, and other one will be the template strand. And the synthesis of RNA is from the coding strand, which will code the different sequences of RNAs. Finally, we will move towards the recognition of termination regions, which are different in both prokaryotic and eukaryotic species. This is, the green one is the structure of RNA polymerase having a lot of components, and it is solely depend upon the RNA polymerase to go and initiate the process of RNA synthesis in the first step of RNA synthesis, which is known as initiation. So binding of RNA polymerase holoenzyme with the promoter region. RNA polymerase will only go and bind with the promoter regions. These promoter regions are the consensus sequences or conserved sequences present near the start of the transcription sites. There are two types of promoter regions present in case of prokaryotic cells. These are known as -35 sequences and Pribno sequences. -35 means these sequences are going to be present at the negative site or prior to the start site of the transcription. And in close proximity or 19 base pairs apart, there is another box or conserved sequences box is present, or a promoter region is present, which is known as the Tata box, Pribno box. The sequences present in -35 sequences are TT, GA, CE, and 19 base pair apart, TA, TAAT are present. If RNA polymerase recognizes both the sequences, what it's going to do, it will go and attach at these sites, which are the promoter regions, and then will start the process of transcription. Here you can see the double-stranded DNA, and in the red color and blue, RNA is going to be synthesized when the RNA polymerase is going to bind with the promoter region. So local unbinding by the binding of holoenzyme with promoter region occurs, which causes the formation of RNA-DNA hybrid helix. So RNA polymerase does not need primer and proofreading activity. As we discussed before, some of the many differences between the replication and transcription process. So initiation is done when the RNA polymerase binds with the promoter region, and elongation is done when there is the start of the RNA synthesis from the double-stranded DNA. Finally, the prokaryotic cells are going to get terminated with the help of different termination sequences in the final step of transcription, which is known as termination. So termination could be of two types. One is known as intrinsic and the other is known as extrinsic, or one is known as dependent and other is known as independent. So intrinsic means there are some special types of sequences present internally at the different sites of DNA which are responsible for the stop, or which acts as the stop codons for the to stop the further synthesis of RNA. And if there are no intrinsic signals are present which are responsible for the termination signals, then some other special types of proteins called as Rho proteins or the Rho factors are there to cause the termination of that particular part of the RNA synthesis. So intrinsic termination could be called as a Rho independent termination because there is no Rho factor or protein is required for the termination of this specific type of termination. While in the other type, which is the Rho dependent termination, special types of proteins known as Rho factor is involved for the termination of this RNA synthesis. Here you can see in this diagram, which is the number 30.9 in your Lippincott transcription chapter, one diagram showing that there is some internal sequences present which are responsible for the termination of the transcription machinery. First of all, see, there is the double-stranded DNA. One strand is the coding strand and the other strand is the template strand. And a nascent RNA or newly synthesized RNA is formed from the template strand of the DNA. So in the nascent DNA, when the termination is going to happen, then some special intrinsic bases are going to repeat themselves, like G, 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 C, U, U, U. If there are repetition of these bases, there will be a chance that G and C will bind together, because G and C bonding, always bonds with cytosine with the three hydrogen bonds, which is a very, very strong bonding. So wherever G finds C, it will go and bind with it. That is why a hairpin-like lobe is going to be synthesized, which causes the detachment of RNA from the DNA. So this is the overall mechanism of your intrinsic termination. And what about the Rho dependent termination? Rho dependent termination requires Rho proteins, which is hexameric adenosine triphosphatase, and ATPase dependent helicase activity. So here you can see a vast function of Rho protein or Rho factor. Rho protein and Rho factor are the same words. While in the previous type of termination, you learned about that the termination is Rho independent termination. We did not need any type of Rho proteins in this diagram, which is the Rho independent termination of transcription. While in the Rho dependent termination, we are going to involve the Rho protein, which is which is having ATPase activity and helicase activity. So this helicase and ATPase activity in combination is going to cause the detachment of RNA from the DNA. So we will be getting the nascent DNA from the nascent RNA from the DNA. This ATP dependent helicase activity releases the RNA. So this is the overall concept behind the two types of terminations, which is the row independent termination and row dependent termination. And this type of termination, which is having many classes and a very strong concept behind it, is very important according to examination. And this is often asked in examination, and it is the favorite part or the favorite question which may be asked in your exams, that is how the termination in prokaryotes occurs. So this is all about today's lecture. From tomorrow, inshallah, we will be discussing the transcription process in eukaryotic system.